Good morning. Welcome, everyone. So it's my pleasure this morning to introduce Dr. Wes Van Voorhis to speak. Dr. Van Voorhis is a UW Professor of Medicine in the Division of Allergy and Infectious Diseases, as well as an Adjunct Professor of Global Health and Microbiology. He serves as the Director for the Center for Emerging and Re-emerging Infectious Diseases, a center which takes a multidisciplinary approach to identifying and developing diagnostic, therapeutic, and vaccine solutions to emerging infectious diseases. Dr. Van Voorhis earned his PhD in immunology at Rockefeller University, where he was the first to discover and characterize human dendritic cells. He subsequently earned his MD at Cornell University Medical College, went on to complete his internal medicine training at UCSF, and completed his fellowship training in infectious diseases here at the University of Washington. As a scientist, Dr. Van Voorhis's research has focused on preclinical drug development for malaria, trypanosomes, leishmania, and cryptosporidium. His lab is now characterizing a new preclinical drug candidate for the treatment of cryptosporidiosis, toxoplasmosis, and several animal diseases. This same compound shows promise for blocking malaria transmission. His research efforts have led to over 260 peer-reviewed papers and numerous academic awards. It is with this background that he comes to us today. Please join me in welcoming Dr. Wes Van Voorhis as he presents Malaria, What's New and What's Needed for Eradication.
Well, thank you very much for that lovely introduction. I hardly recognize myself, but I just want to point out I do have a conflict of interest. I spun off a company called Paratherapeutic through University of Washington CoMotion, which is developing bump kinase inhibitors, a drug I'll mention, but for animal health, not for human health, where I'll mention its context today. So today, I was asked to give these grand rounds about 4 years ago to bring back some light from a sabbatical I did at the Medicines for Malaria venture, which I'll tell you about. That's a public-private partnership based in Geneva. Bill was kind enough to let me go for a year while I was division head. I'm forever grateful to Jeannie Marazzo. Unfortunately, Jeannie learned to love to be a division head and went off to University of Alabama and is doing a great job in that and competing with us. But more about that later if you wish. So what am I going to cover today? I'll start with the traditional case, move to what's going on in terms of the epidemiology. You may have seen the headlines that malaria elimination is being stalled, or malaria control is being stalled. The clinical characteristics of malaria, what's helping now to control malaria, what's due to the reduction we've seen in the last 15 or so years, what are the new dangers to our control efforts, and what's really needed for eradication. And by eradication, I mean what Melinda Gates and Margaret Chan announced in 2008, that they wanted to push the world towards a new attempt to eradicating malaria, similar to how smallpox has been eradicated from the globe. And we'll talk about the technological advances that are coming forward to help with that now possibly in the year 2040 is what's projected. So first with a case, a 3-year-old Ugandan boy felt bad at home, had chills but was warmed at the touch. Later he became increasingly sleepy. His mom brought him into a local medical clinic where he was unarousable and high fever. And this is the face of cerebral malaria, which kills about half of the children between 1 to 5 years old, particularly in sub-Saharan Africa, where our children are particularly vulnerable to this. Malaria the word is derived from the Italian mala aria, which doesn't mean bad opera, but actually means bad air, and was named that because of the areas around swamps and so on where it was felt to be precipitated by the evil humors from the air, but Laveran in the French Foreign Legion discovered malaria was a parasitic illness that resided in red cells in Algeria in 1880 and later won the Nobel Prize for that in 1907. Ross subsequently discovered in India that malaria parasites could be found in the Anopheline mosquitoes, explaining the wet association and mosquito growth in wet areas, winning the Nobel Prize in 1902. And these fundamental observations led to understanding and control of malaria and elimination from some places, such as endemic transmission in the US. So this is what's happening with malaria burden since 2000. Malaria burden actually peaked in the 1980s or so, and then a concerted effort was really started around 2000 to bring down the number of deaths shown on the right with the red line, and the overall incidence, which climbed until the year 2003 and then gradually sloped down. What's alarming people a little bit is that the malaria incidence has been trending up slightly in the past 3 years, as you can see in the red box with the blue line. And that may be a little bit false because of population growth. The incidence really hasn't changed. There's still the same number of cases per 100,000 that are at risk. But nonetheless, we are losing the battle on the trend towards keeping the number of cases going down. 93% of the cases are in sub-Saharan Africa. As you can see, this picture from Lou Miller, science article with the Anopheline mosquito casting the dark shadow over sub-Saharan Africa, leading to the most deaths there. And the most vulnerable really are 1 to 5-year-old children and first or second time pregnant women that have the most morbidity and mortality associated with that. In fact, the majority of the deaths are in the 1 to 5-year-old African children. So you may have seen, if you're fans of Gary Larson, even though he's retired, you may have seen this cartoon, which he says he got the most mail from of any cartoon, but it wasn't fan mail. It was mail from junior entomologists who questioned the validity of this. Can you see what's wrong with this? Yeah, it's the male mosquito that comes in and says, what a day, I must have spread malaria across half the country. And every junior entomologist knows that the female mosquito must get the blood, and the male just goes around drinking pollen from and sugar water. So this is the malaria life cycle. It starts with the malaria mosquito taking up sexual forms, and about 10 to 14 days later, it bites another human. At that time, it needs more blood, and it will be infected with the malaria after undergoing the sexual cycle in the mosquito. The mosquito transmits from its salivary glands the sporozoites, which travel through the bloodstream in an amazing journey to find Kupffer cells and eventually hepatocytes in the liver. And there they grow and rupture most times within 5 to 10 days. But for Plasmodium vivax and ovale, you may remember that it can sit in a dormant form in the liver for up to 4 to 5 years and then emerge at a later time, probably helping overwintering in temperate zones where mosquitoes are not always available. But in any case, you aren't symptomatic with the few sporozoites that come into the liver, but you are symptomatic when the merozoites emerge from the liver cell, the infected liver cell, and infect the red blood cells. And it's the intraerythrocytic cycle that you can see here that leads to most of the symptoms, as there's amplification of the malaria during that time. And particularly during the rupture of these merozoites and release of so-called endotoxins that lead to fever and muscle aches and the classic symptoms of malaria is where you get the particular disease. At some rate, the trophozoites form these gametocytes, which are the infectious forms for malaria that you need to swallow, or the mosquitoes need to swallow a male and a female gametocyte to have the sexual cycle go forward. That happens in their stomach, and parasites move over 7 to 10 days to the salivary glands. So most One thing about the life cycle I want to tell you that will presage the rest of the talk is that most current anti-malarial drugs only act on the intraerythrocytic life cycle, leaving people still vulnerable to acquire new malaria while they're taking those drugs. And for the dormant forms, the hypnozoites, to remain active in the liver. And most anti-malarial drugs don't address the gametocytes, so the persons that are infected after taking therapy may be infectious for a month to 6 weeks later, allowing transmission to go forward. I'll talk about the current malaria vaccine that's been approved by the EMA that acts at the sporozoite level. It's a split vaccine that blocks sporozoite infection. And I'll talk about insecticide-treated bed nets and indoor residual spraying that obviously act to keep mosquitoes away from completing the life cycle with humans. So malaria signs and symptoms are probably aware to all you guys, and you probably ask where people have been when people come in with fevers. You should be asking, even in the last 4 or 5 years, have you been to malarious areas or had any malaria risks? And people generally have repeated high fevers. They may not become synchronous, as is commonly thought, especially in non-immune people like you and I, but maybe hectic fevers. They have regular shaking chills, sometimes an enlarged spleen, reduced red cells and anemia, muscle pains, and then the potential fatal complications include severe anemia, which kills about half the children in Africa, and cerebral malaria, which kills about the other half. Shock and low blood pressure come along with the malaria rupture, and lung, kidney, and liver damage can happen partly because of shock and partly because of their propensity to stick in the microvasculature, causing clogging and poor oxygenation. So how do you remember the signs and symptoms of malaria? I'll give you a mnemonic. Malaria, I just got a chill from malaria, and suddenly I knew a fever would come too, and sweat. Malaria, I think I shall die from malaria. My breathing labors on. My consciousness is gone, I'm pale, malaria. Sing it soft and you'll hear skeeters whining. Sing it loud and your spleen will be pining. Malaria, 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 malaria. Thank you. Most medical students I've taught can't remember my name, but they can remember my songs. Anyway, what's really helping us to control malaria now? It's a combination of vector control and improved drugs. And you probably know about the President's initiative to address malaria, TB, and AIDS. And they have been helping fund this, as well as countries around the world. There's residual spraying that goes on in houses such as shown here that kill the resting malaria females, insecticide-treated bed nets that you can see are being implemented around the world, and improved drugs such as Artemisinin combination therapy have led to this downslope in incidence and this dramatic downslope in the number of deaths from 2000 to 2015. So first, insecticide-treated bed nets. This is one of the most cost-effective public health interventions. There have been greater than 20 field trials in children, and with this increased coverage that's occurred from the year 2000, shown here in red, where basically 0% of the sub-Saharan Africa population were sleeping under bed nets, to the current situation here in 2012, where up to 100% of some countries and regions are sleeping under insecticide-treated bed nets. This has led to a 35% estimated reduction in malaria deaths and dramatic reduction in the incidence. And if you're interested, donating $10 to Nothing But Nets, which is a WHO nonprofit, helps deploy a single net. I donate to this every year. Malaria drug therapy got better. As you probably know, there was resistance that spread throughout the world on the older drugs, chloroquine and Fansidar, sulfadoxine pyrimethamine. And shown here are dots where there's resistance described in 2002, basically making these drugs largely inadequate for most of the regions where malaria is spread. And then along came Artemisinin-based drugs. And there's a long history of Artemisinin. Artemisinin was first known as a tea or an extract that was made from Chinese wormwood, or Artemisia annua. And it was known since 200 BC to abate fevers in China, particularly in southern China where malaria was endemic. And the Chinese scientist Tu Youyou isolated Qinghao Shao, or Artemisinin, from the plant in 1972. This actually helped their efforts with malaria control and was shipped to Vietnam and strategically used against us, if you will, during the Vietnam War to have better malaria control than we were having with our soldiers because of the resistance there. So she had derived that, and that was adopted, and the Nobel Prize was given to her and to other scientists that derived another breakthrough antiparasitic drug called ivermectin in 2015. So artemisinin combination therapy is the combination of artemisinin, which is fairly short-lived in blood, but leads to a dramatic reduction, rapid reduction of malaria parasites with a longer-lasting partner. This is given usually over a 3-day course, once to 3 times a day, with a bunch of partner drugs. Here is shown the one that Novartis formulates that's currently approved in the US, lumefantrine. And this combination adds to efficacy in completely eliminating the malaria from the blood, because a single dose of artemisinin, though you feel better, the parasites would generally relapse. Using 2 drugs at once, you can imagine that there's a much lower chance of emergence of resistance. So artemisinin combination drugs have really been the drug of choice in Africa and Southeast Asia, and have been universally adopted for their use by departments of health in those countries and the WHO. So what are the new dangers, though, to the progress we've made? One of those is, I mentioned insecticide residual spraying and treated bed nets. The treated bed nets in particular, and to some degree the majority of the IRS or insecticide residual spraying, depends on this single insecticide, pyrethroids. And pyrethroids are emerging as a major source of resistance in mosquitoes. You can see here in 1995 to 2000, there was still quite a bit of sensitivity in the green dots for pyrethroids, but now in the latest survey in 2008 through 2014, pyrethroid resistance has spread and become almost universal throughout sub-Saharan Africa. The dark spots are where malaria is most endemic, so you can see the overlap is very concerning. And then this leads to less efficacy of the bed nets and indoor residual insecticides. So that may be one reason we're seeing a flattening of the reduction of the incidence of malaria. Another reason is perhaps resistance to artemisinin combination therapy, which I told you about. So this was the first report in the New England Journal, and it's become widespread throughout Cambodia, Thailand, Myanmar, that artemisinin resistance is on the rise. And this was due to the observation of very astute clinicians who noticed that even though in the test tube it appeared that they hadn't lost efficacy for artemisinin because of an artifact of the way it was tested, that clinically it just wasn't working as well to lead to the dramatic reduction that was normally seen with artemisinin. So I mentioned that the breakthrough of artemisinins is they rapidly lead to the reduction. This is a log phase reduction in the number of parasites in the bloodstream normally. And other anti-malarial drugs lead to slower clearance. Well, what's happened with the emergence of artemisinin resistance malaria in Southeast Asia is there's delayed clearance. And this was first recognized in the western border of Cambodia by Francois Nostin and colleagues, and later was linked to a genotype called Kelch13, which is a propeller type transporter that actually pumps out artemisinin out of the malaria cells. So this is necessary for the artemisinin resistance phenotype, and this has led to delayed clearance of parasites and worse outcomes. So what do we need to do for the eradication campaign that was announced by Melinda Gates and Margaret Chan of the WHO? Well, really, we need to improve our tools, the drugs, vaccines, vector control, and diagnostics. And what's really been driving this is product development partnerships, such as Medicines for Malaria venture I'll describe in a moment, that help drive the progress in this area. And what we need for drugs is to address artemisinin combination therapy, that resistance that's popped up in Southeast Asia and probably will spread into Africa soon, although it hasn't at this point. We need to address the liver and transmission stages that I mentioned on the life cycle that are unaddressed and allow continued transmission and reemergence of malaria after people are treated. We need better vaccines for prevention, and I'll talk about what's available now and what's coming along. We need better vector control with new insecticides that address the resistance, and possibly combination insecticides that I'll talk about in the same way that we use combination drugs. And there's a emergence of technology in genetic alteration and gene drive in mosquitoes that I'll talk about that many are excited about for the control of the vectors. And then finally in diagnostics, I won't talk about this greatly, but rapid diagnostic tests, or RDTs, like is shown in the bottom corner there, are available now. They're just not very sensitive. They can pick up people that are symptomatic with malaria, but it turns out that up to 30% of malaria transmission happens in people that have low-level parasitemia that these rapid diagnostic tests won't pick up. So nucleic amplification technologies such as PCR and so on are under study for the eradication process, and Sean Murphy in our Department of Lab Medicine helps lead the group on that. That'll be necessary to find pockets of where transmission is going on even after you've eliminated the symptomatic people. So let's talk about drugs a little bit. So Medicines for Malaria venture, I went to a very scary malaria region called Geneva, Switzerland, to work at this place. It's a Swiss foundation that has partners in over 50 countries and is doing everything from clinical trials to early development. They're funded by the Bill and Melinda Gates Foundation and many governments and even oil companies and places that are concerned about malaria. And they have the largest pipeline of anti-malarial drugs, over 50 products from discovery, clinical trials, and creating access to anti-malarials. They're a successful partnership model, and their efforts have led to 11 approved products. So I'll jump to the pipeline of Medicines for Malaria Ventures supported projects shown here, and these stretch from the early research to translational, moving into human volunteers, to product development in the patient exploratory and confirmatory phase 2B and phase 3 trials, to regulatory approved and access. So just to highlight a couple of things, recently approved by the EMA was tefenaquine, which replaces primaquine. You probably know about, you may know about primaquine, which is used to treat the hypnozoite form of the liver in Plasmodium vivax and ovale. Unfortunately, you can't take primaquine if you have a severe reduction in the glucose 6-phosphate dehydrogenation. So they found an analog, GSK found an analog and developed it with MMV called tefenaquine. The breakthrough here is that you can take a single dose of tefenaquine and cure the hypnozoite forms out of the liver. But unfortunately, it still has the side effect of the glucose 6-phosphate dehydrogenase deficiency. So it's being distributed with a rapid diagnostic test to find out if you have G6PD, so it can be used. But newer drugs that MMV are now delivering have the promise to eliminate this form along with the therapy, and hopefully will not have the G6PD issue in the future. Another drug I want to point out, given the case I gave you, is rectal artesunate. Rectal artesunate is specifically formulated for children who have cerebral malaria and are unable to take oral medications, and it's being distributed to low-level centers around the areas where malaria is hyperendemic for children, like I mentioned in the original case, and they can have a suppository put in and then be transported for more advanced care to a tertiary care center where they can deal with things like intravenous lines and so on. And these have been absolutely life-saving in terms of those children, because the artesunate as a single rectal suppository drives down the parasite level to manageable levels, and the child really most of the time wakes up by the time they're at tertiary care for the final treatment. But what I was really excited on and working with is new drugs in the research and development pipeline that address the Artemisinin combination therapy. And all of these drugs from here on down address Artemisinin combination therapy. They've been tested in volunteers and also in vitro, and found to get over that lesion, give rapid clearance, and treat. And many of these shown with this form or this form act in the liver or stop transmission, work on the gametocytes. So you can see, such as this agent, MMV-048, would provide those lesions for the current Artemisinin combination therapy, addressing the gametocytes and the liver forms. And the target of all these drugs is to make it a single dose, because it's well known that Artemisinin combination therapy is taken in 3 doses, and most people don't take 3 or more doses of the medicine. They feel better after taking 1 or 2 doses, often thought to drive the emergence of resistance. So I want to talk about the patterns they use to find new drugs. This is one pattern where they took the model, the existing template of Artemisinin as shown here, and a chemist by the name of Jonathan Vennerstrom started working on endoperoxides. You can see the endoperoxide lesion and linkage in this molecule. And they found more and more stable endoperoxides until they have this drug, which is now currently in phase 2B trials with a combination drug called OZ439 or artefunumab. And it's been a breakthrough medication, and it addresses the Artemisinin combination therapy, even though you can see it has a vague resemblance to all the oxygens that are in Artemisinin. Does not seem to be susceptible to the same resistance mechanism. Another way they do it is by molecular design. So dihydroorotate dehydrogenase was known for some time to be a good target, and our UW chemistry professor Pradeep Rathod, and I believe Fred Buckner had a role in some of these experiments. Pradeep developed their initial hit into a drug that's now being used in phase 2A trials in Africa and South America, called DSM 265. And this group has developed a very valuable drug that is not rapidly clearing of malaria, but it will be a nice combination for a drug like I showed you a moment ago, and addresses the liver forms as well as potentially the gametocyte forms. And then finally, I like this term, ask the parasite. I was part of helping coordinate acquiring new chemistry from drug companies and from academic groups, and testing those against the parasites using high throughput screening, or HTS, using robotics like is shown here, to develop hits, and then use medicinal chemistry to develop leads that lead to new candidates. And there are now dozens of new candidates that came out from these high throughput screens that are under advanced testing. They screened over 7 million compounds and have 25,000 sub-micromolar hits. And these actually, you could select for resistance in vitro, and with the majority of drugs, they were able to detect the molecular target by whole genome sequencing and finding repeated mutations in key parts of these molecular targets for these drugs. A short list is shown here in an effort led by Elizabeth Winsler of UCSD. And we at the University of Washington, along with Center for Infectious Disease Research at Seattle Children's and UCB Beryllium, are using structure-guided drug discovery to help find new scaffolds for these various targets. And I won't talk about that further, but there are many efforts going on in the early medicinal chemistry in this area in Seattle. One thing I did want to talk about that KK or Kyoto Ojo has been pushing on. Kyoto Ojo, in case you don't know him, is an associate professor of medicine here at University of Washington. And Kyoto has experienced malaria more times than he can count. And he became very interested in the bump kinase inhibitors and their effect on the calcium-dependent protein kinase shown here, moving as it encounters calcium to activate it to become an active protein kinase. It turns out when you specifically block that calcium-dependent protein kinase, the gametocytes that are swallowed by the anopheline, the female anopheline mosquito, are unable to undergo their sexual cycle and become infectious. So what actually happens is this CDPK4 is a necessary trigger for the emergence of these sperm-like molecules called microgametes that have to infect the microgametes to become the egg part or the zygote. So in honor of Bill's efforts in the male, stopping the males from becoming fertile, we call these a molecular condom for malaria, and the sperm are not actually able to make it to where they want to be able to complete the life cycle. A mosquito doesn't get infected, and our vision is, or Kyoto's vision is, that we now have a compound that is available for at least a couple of weeks as in the mammalian bloodstream, can stop transmission, and that may be sufficient to make a major impact in terms of the transmission of malaria. So he's co-developing this with the Medicines for Malaria venture, and hopefully it'll become a useful part of the armamentarium. It's interesting in that unlike the other drugs I showed you earlier, it doesn't have any effect on the asexual cycle that actually causes the fevers and so on. But that may be a good thing because there are very few gametocytes, so emergence of resistance is less likely to occur compared to the billions and billions of asexual forms that are found in the bloodstream. Okay, so one thing I've talked to you a little bit about malaria drugs is combination therapy is a desirable thing. And so how do you find the right partner for the drug you have to treat the right population and so on? Do you pick a trusted friend, an old malaria drug, or a beautiful stranger, one of the new ones under development? And one of the things I did at the Medicines for Malaria venture was study all the different partners that were available and come up with algorithms for this, because as you can imagine, this is a fraction of the malaria pipeline shown here, older, newer, and newest drugs that are available for combinations. If you take a 2x2 combination of all of these, you have thousands of different combinations that could be put into human trials, and that's just not possible. So what we did was we took all the available information in vitro, in mouse models, and sometimes available from early human studies in terms of the developmental stages that it hit, the type of resistance, that we wanted single dose to work, mechanism of action to block emergence of resistance, and all these other kinds of characteristics, and ended up with a numerical score that allowed us to pick relatively few combinations to go on into the mouse model of malaria, the severe combined immunodeficiency mouse model that uses human malaria, and then ultimately into the human challenge model, which has been used to a great benefit by Medicines for Malaria venture to characterize drugs. And this boiled it down into a reasonable number of combinations that are moving forward now into clinical trials. I also wanted to do a call out to Carol Sibley, who co-founded the worldwide Anti-Malarial Resistance Network, and their mission has been gathering and providing information on molecular and phenotypic markers of resistance to help slow and manage the anti-malarial drug resistance that's happening around here. And this is a screenshot from her website showing, I mentioned lumefantrine as one of the artemisinin combination therapies. This is showing a marker for a reduction in the rate of efficacy of lumefantrine throughout Africa. And as you can imagine, the bad ones are the red dots, where they're finding more and more lumefantrine failure, if you will, compared to other parts of Africa. It doesn't mean that it fails all the time. It moves from approximately 99% success to about 85% success, but that's still a substantial problem. So Carol and her colleagues are now doing a clinical study looking at combining lumefantrine with another drug called amidiaquin and artemisinin. The two have opposing selection for resistance mechanism, and it's thought that by deploying all three drugs at once, that emergence of resistance won't occur at all. So that's under trial now in Africa. And then now I want to switch to talking about the malaria vaccine. So as you probably haven't heard a lot about the malaria vaccines, except to know recently the EMA has approved one, and that's the RTSS that's being rolled out now, and I'll tell you about the rollout in pediatrics. So RTSS is a pre-erythrocytic vaccine, meaning it works on the sporozoites in the liver before the erythrocytic cycle. There are also some transmission blocking vaccines that are they're getting favorable results. These block the gametocytes from being able to infect the mosquitoes. They don't actually protect the actual person that had the vaccine, but they stop transmission in the community. And there's to date, the blood stages vaccine that work actually on the intererythrocytic cycle have not emerged into the forefront because of issues with antigenic variation on the blood stages. But this vaccine, Mosquirix, or many of us still refer to it as RTSS, AS01, has emerged as an effective vaccine. It's been over 30 years in development. It first came from observations from Ruth and Victor Nuzenzweig in mice that the sporozoites, if they could be inactivated by radiation, could be protective against acquiring malaria. And they characterized the circumsporozoite surface antigen, or CSP, which has repeats that are in the vaccine, and it's combined with an adjuvant that was developed by Steve Reed, who runs Infectious Disease Research Institute here in Seattle, and the specific adjuvant was just as important as the specific immunization in the thing. The EMA, as I said, has basically approved it since 2015, and it's being rolled out in the malaria vaccine implementation program by the World Health Organization, by the Program for Appropriate Technology in Health, or PATH, and GlaxoSmithKline that helped co-develop it. And they're now vaccinating 360,000 children a year in Ghana, Kenya, and Malawi. And there are still challenges to this. In the initial studies, the protective efficacy was only about 30 to 40%. And you might say, well, that seems pretty low. Why are they rolling that out? There's still a projection that 100,000 children's lives can be spared a year at the current efficacy. Cold storage is required, but there's that issue with other vaccines. And the greater than a year protection requires 4 inoculations. But a new study that's promising that came out of Walter Reed at the American Society for Tropical Medicine and Hygiene this year reported that fractionated subcutaneous dosing led to enhanced protection, moving the protection in adults from about 50% up to greater than 90%. So this is being rolled out in Africa in small children now, and hopefully the fractionated dosing will lead to even more enhanced protection. But what's coming along is very interesting. I mentioned that sporozoites could be injected and protect people. That was shown in human volunteers by irradiation of sporozoites. Stefan Kappa and co-workers at Seattle Children's Research Institute or at the Center for Infectious Disease Research, or former Seattle Biomed, have developed a genetically altered parasite with a triple knockout. And what happens is that when they inject the sporozoites, they invade the liver cells and arrest development. And that leads to a small amount of parasitic antigen that leads to protection. And they're still working on another generation of this where there's a replication competent genetic altered parasite that will go on to produce more antigens. But they've already moved the triple knockout, the one that delivers to mosquito bite administration in collaboration with Jim Kublin at the Malaria Challenge Center, and showed that this is safe in humans. It doesn't lead to actual malaria, but does give protective type immune response. And now there's a preliminary efficacy study going on. The vaccine is being bottled by a company known as CERID that has worked out the kinks in terms of bottling sporozoites for effective vaccination and efficacy assessment with homologous and non-homologous malaria challenge in human volunteers will occur in the next year or two. So we're very excited about that possibility. And if you want to acquire malaria here in the US, you can talk to Jim Kublin and colleagues who work at the Fred Hutch, Cider, Scree, and UW to do the Malaria Clinical Trial Center. They're actually raising lab-reared Anopheline female mosquitoes, which can feed on your arm and give you malaria, and then you can be treated with an experimental drug or a vaccine. Doesn't that sound like fun? The best part is they monitor you very closely, and not a single volunteer has actually gotten clinical malaria. They're able to detect it in the blood by PCR before anybody gets sick. So I'm going to end up with vector control for eradication. I mentioned the pyrethroid resistance as threatening vector control, and so novel insecticides are being developed. And then there's genetic engineering of mosquitoes that many people are excited about. So there's another product development partnership group called International Vector Control Consortium, and this is their product development pipeline focused mainly on insecticides. And they have novel insecticides under research and development. They have new bed nets that have introduced 2 novel insecticides at once. And these, though they're more expensive than the previous pyrethroid type bed nets, they're being accessed and launched. They've already driven the cost down from about $50 a bed net, which will last for 5 years, to about $15, and the cost is coming down even more. So this should address the issue with insect resistant bed nets. There are novel insecticides now that are being introduced 2 by 2, again to try and stem resistance for indoor residual spraying. And there's even novel outdoor approaches where they use attractive sugar bait to bring mosquitoes in, and those have been used in field trials and shown to actually reduce the incidence of malaria. And then I'll end on genetic approaches to control malaria. These are the various approaches one can imagine, and there's various experiments going on in all of these stages, including interfering with the survival of reproduction, which should reduce the numbers of mosquitoes, potentially eradicating or leading to species collapse, biasing the sex ratios towards males, which again would leave the males kind of lonely and lead to a population collapse. Changing the female's host preference to feed on non-humans from human preferences, and this should lead to reduced malaria, causing the death of infected female mosquitoes, so specifically targeting where the problem is, and modifying the females to no longer support parasite development. And there are a series of genes that can be altered that can give these various phenotypes. So Bill Gates at the World Health Summit in Berlin in November got up and said for the first time he didn't feel bad about perhaps a given species being driven from the face of the world. Though it remains controversial and a lot of environmental studies need to be done to implement these sorts of strategies. But you probably heard this term gene drive, and I wonder how many of you understand what's going on. Gene drive is where genes are inherited at greater than a Mendelian rate. So this is, you know, a traditional Mendelian transmission of 2 genes where there's a 50-50 distribution, and the relative frequency of the genes remains constant over time. With gene drive, there's a proportion greater than 50% that drives that gene into the population. So in this example, at a 70% driver, over about 20 generations, you reach saturation where almost 100% of the population has those particular has that particular genotype, and it can be actually a much faster driver where if D equals 100% and there's no fitness effect to the mutation, then the frequency can go from 1 to 99% in only 9 generations. Given the mosquito generation is about a month, that means in 9 months a population theoretically can be completely replaced. So how do you do gene drive? Well, you probably all have heard about CRISPR-Cas9, the babies, the twins that supposedly were genetically altered in China this year. CRISPR-Cas9 is this system of nuclease targeting where there's a specific guide RNA that drives it to a specific gene sequence. Cleavage occurs because of the Cas9 effect, and so there's nuclease and specific targeting. And this has been being used now in gene drive mechanisms where a gene can be introduced with the CRISPR-Cas9 nuclease and a germline promoter to knock out a gene or to drive in a new gene function, for instance, to add a sense of smell where mosquitoes would prefer cattle over, say, humans. And then gene drive can also be used, for instance, to be located on the Y chromosome to recognize repeats in the X chromosome, leading to multi cleavages in the X chromosome, functionally favoring the Y chromosome sperm, leading to bias the sex ratio towards males. So all of these approaches together are being explored. There are some field trials with several of the approaches, small field trials that have been done under contained situations. But as you can imagine, the world scientific community is taking a very cautious approach to the total rollout of these gene drive approaches in a population and trying to understand what could be the environmental impact of such an intervention. So in summary, I've told you about the advances in malaria technology that are important for the eradication agenda. In vector control, now we have insecticide-treated nets that last 3 to 5 years. and residual spraying. Unfortunately, emergence of resistance to the major insecticide that's used, the pyrethrins, has come, but in the future and now we're rolling out novel combination insecticides, including effective bed nets that have 2 effective new agents. And we have gene drive to experiment with in altering the malaria mosquitoes. Vaccines include the now available Mosquirix vaccine, which gives us about 30 to 40% protection. But in the future, split dose enhancement will be looked at to hopefully drive the efficacy to be better. And live vaccines are under development, like that being done by Stefan Kappa at the Center for Infectious Disease Research, Seattle Children's Research Institute. And then in drug development, we have Artemisinin combination therapy, which is still largely working in Africa, not so great in Southeast Asia, but there are future new therapies that are emerging from PDBs like the Medicines for Malaria venture that act on transmission and liver blocking drugs and address the Artemisinin combination therapy. So I want to thank Tim Wells, who is the Chief Scientific Officer at Medicines for Malaria Venture, who helped me update this talk, Sally Ethelston and Dave Kaslow at PATH Malaria Vaccines Institute that helped me with that part, Nick Heyman at the International Vector Control Consortium, and Stefan Kappa at Seattle Children's Research Institute. All of you for your attention, and Bill for allowing me to go for my sabbatical year. Thank you very much.